I am excited to share a little bit about our journey in learning how to optimize the way we approach studying the urobiome. Before I get started, I wanted to recognize the people who really contributed to this work. So I'm sharing a study that was recently published from our lab. This was started by Angela Scott several years ago when she was an undergraduate in my lab. She's now a third-year vet student, and it really grew to another level through Zachary Lewis's master's degree, and I'm going to share that with you now. So the urobiome is the microbiome of the urinary tract, which can include the kidneys, ureters, bladder, and urethra. And if you're here at this meeting, it may come as no surprise to you that healthy urine is not sterile. And this has been well established in a number of ways, including through both culture and sequencing. I won't get into that at the moment, but as so many of us are interested in knowing, we want to be able to ask questions about, is the urobiome driving diseases like bladder cancer, incontinence, urinary tract infection, urolithiasis? These are all conditions that have been associated with shifts in the urobiome. And so really we want to go to the next level to try to understand urobiome function and how it may be driving or underlying some of these diseases. But one of the challenges with studying the urobiome is that it's low microbial biomass and can potentially have high host biomass. So for example, if this is a urine sample, here are the little squigglies in red and purple, those are microbes, but within urine, there's not that many, particularly in healthy urine, there's not that high of a microbial biomass. And then you also have host cells. So here are these big purple circles. Those are your host cells. So whether it's human host cells, or if I work in dogs and I'm looking at dog host cells, they've got a lot of DNA. They can dominate in things like shotgun metagenomics, or they can bias 16S kinds of reads when we try to profile the urobiome. So my lab really tried to figure out the best ways of studying the urobiome. And the goals of the study that I'm sharing with you today were, does urine sample volume impact urobiota profiles? Do host depletion methods affect urobiome profiles? And can we reconstruct metagenome-assembled genomes of urine microbes from shotgun metagenomic data? And I'll give you a heads up now, we used dog urine for this. So a quick view of the dog urinary tract. Here's females, this is bladder and urethra. Here's males, bladder and urethra. And so the first question we wanted to ask was, does urine sample volume impact urobiome profiles? This is because as we looked at the literature, studies reported using anywhere between 0.5 mils of urine and 50 mils of urine, which is a huge difference. So we were trying to figure out how much urine should we be using for a good and reproducible study of the urobiome. And because this is a very short talk, I'm going to give you the spoiler right up front. We took dog urine from 5 healthy dogs. We aliquoted it into fractions ranging from 0.1 mil up to 5 mils. And then we looked at it using 16S sequencing. And we found you should use at least 1 mil. Again, this is healthy voided urine from dogs. But the greater the volume, so 3 mils or more, really produced more reproducible and consistent results. And so that helped us know something about volume. What that looks like here, so here we've got a Bray-Curtis plot. Shapes are the dogs, individual dogs, and colors are the volumes, with darker colors representing larger volumes. You can see these samples are really clustering by dog, and so inter-individual differences were really overwhelming differences due to volume. So that was a good thing for us to know. But you can also see here on the y-axis, we've got unique ASVs, or richness, microbial richness. And you can see that that richness really increased as that volume increased, which helped us know that we should be trying to maximize volume when we're collecting urine for studies. In the second study that we looked at host depletion methods. So urine does contain host shed cells. And in some cases, things like inflammation or infection or bladder cancer, there may be a higher load of cells shed into the urine. So we took urine from 7 healthy dogs, and we tried extracting it across 6 different methods, 5 of which included host depletion steps. We also spiked that urine with additional host cells, so we knew exactly how many host cells were going in through our spike. And so here's our urine sample again, and our goal was to try to get rid of host cells. So our Fast takeaway on this was, yes, all of these methods did affect the urobiome profiles. If you are expecting low host biomass, we were most successful with the Bacteremia kit. And if you are expecting high host biomass, we did best with the DNA Microbiome kit. And in that case, we were able to get the highest microbial diversity and lowest abundance of contaminant reads and greatest number of metagenome-assembled genomes with that DNA Microbiome kit. And again, the inter-individual differences overwhelmed differences due to extraction method. So again, the colors here are dogs and the shapes are extraction methods. And here, DNA microbiome is really the one I want to call out. So microbial diversity is on your y-axis, and DNA microbiome kit had the greatest microbial diversity. And we're using that as a proxy of a way of really capturing rare species. The last thing we asked was, can we actually reconstruct metagenome-assembled genomes from urine microbes and shotgun metagenomic data? And the answer was yes, but we had limited success with this. So we generated 26 total NAGs, 11 were from our positive control, 10 were probable contaminants, which really tells you something about how many contaminants are in low biomass types of media, and 5 were derived from urine, and those 5 are listed here. In one of them, we were able to identify the full pathway for long-chain alkane utilization. And here, one of my other interests, bladder cancer, is associated with environmental chemical exposures. And so I really wanted to look into these microbes to see, can we see environmental chemical metabolism? And the answer was yes. And this is just a proof of concept. It's something that we can see. And that's important for me to be able to understand, to say, how could these urine microbes be contributing to something like bladder cancer? So our final takeaway here is, if you're venturing into the urobiome world, you really want to try to maximize your urine volume to increase the reads and improved metagenome-assembled genome recovery, because understanding function is going to be really key to understanding how urine microbial communities contribute to health and disease. And this is my crew, and I'm really grateful for this wonderful group that I work with. These are the co-authors on this study. This was supported by the NIH as well as the College of Veterinary Medicine and the Infectious Diseases Institute at Ohio State University. Thank you so much.